My name is Ross McGilvray, and I knew Earl Davie because I was a postdoctoral fellow with him in Seattle from 1977 till 1981. At that time, Earl gave me a recommendation for a faculty position at the University of British Columbia, just north of Seattle. So I moved to UBC in 1981 and stayed there until my retirement in 2017 as a professor of biochemistry and molecular biology, and also as the founding director of the UBC Center for Blood Research. So I went from my PhD at the University of Miami to Seattle in 1977. Earl at the time had a very large group of postdocs, students, technicians. He had one of the largest grants out of NIH for an individual, and he was just at the start of in the middle of purifying clotting factors. So back in 1962, Earl worked with Oscar Ratnoff in Cleveland and came up with an idea for a cascade of reactions or a waterfall of reactions involving the blood coagulation proteins. He built on this over the next few years, moving to the University of Washington. And there he got together with Eddie Fisher, another professor of biochemistry, and they put together a big, large-scale preparation room where Earl could purify large amounts of bovine blood clotting proteins, and Eddie Fisher could purify large amounts of rabbit skeletal muscle kinases and phosphatases. So in an amazing coincidence, you had side-by-side labs. Earl's was involved with control of blood clotting using proteolysis, an irreversible type of control, whereas Eddie Fisher was involved in the reversible control of metabolic reactions by phosphorylation, dephosphorylation. So it was an amazing department back then. I joined a group that had so many distinguished people in it, Walter Kissel, Ko Karachi, Gordon Vihar, Sandra Deegan, Bill Canfield, Kazuo Fujikawa, Doug Malinowski. There were many, many of us all working happily with Earl. So when I think about Earl, I really have four characteristics that I think he passed on to me and to other people in his lab: a sense of collaboration, a sense of collegiality, a sense of scientific integrity, and then a sense of loyalty. So I'd just like to expand on a few of those. Earl's lab, when I entered it, was just completing the purification of many bovine blood proteins. This would involve taking garbage cans full of blood, passing the blood through cream separator, and then purifying the proteins from the plasma by a combination of precipitation and chromatographic methods. In this way, they were able to purify large amounts of protein and also to a very high degree of purity to make sure that the enzymatic activity was attributable to the protein that had been purified. When I entered his lab, Earl was just getting started in recombinant DNA research, and through another collaboration with Savio Wu at Baylor College, we were able to start cloning clotting factor genes, and the big recombinant DNA revolution allowed many, many further studies, which Earl's lab continued, as well as other labs over the next 20 years or so. This included the evolution of the clotting proteins, the clotting genes, determining the molecular basis of bleeding disorders such as hemophilia, the expression of recombinant proteins, and Earl was particularly notable in collaborating with Novo Nordisk on the production of niastase, or activated recombinant factor 7, and then also the mutagenesis of clotting proteins to allow structural functional studies. So getting back to my 4 characteristics of Earl: collegiality. He was just the most charming, caring, and compassionate person you could ever wish to meet. When my wife and I arrived in Seattle, I think we had $100 to our names, so Earl lent us some money, and my wife was particularly anxious for us to repay it, and we repaid it within a couple of months. And Earl said, "Oh, I'd forgotten that I'd actually lent you that money." I also had a visa problem in Seattle, and Earl went to the federal building on my behalf in downtown Seattle and spent 3 hours working his way up the immigration bureaucracy, eventually leading to me being given a new visa and a written apology from the US Immigration Service. Anybody that has interacted with the Immigration Service knows that it takes real charm to get that accomplished. Earl also taught me that research can be fun and that you can be a nice guy and still be a competitive researcher. Earl's friendliness and congeniality extended throughout the international community. And indeed, here at UBC, we initiated some Earl Davie lectures, and we unashamedly used his name to attract the real leaders in the field of thrombosis and hemostasis to come to Vancouver and to meet with Earl and discuss their latest research. This included people like Harold Roberts, Evan Sadler, Ken Mann, Chuck Esman, and more recently, David Lillicrap and Jeff Weitz. Many, many people really admired Earl. His scientific integrity meant that his work was of the highest quality, published in the best journals. He's trained many, many people over the years, and I think he's had such a high degree of loyalty from people that trained through his lab. Importantly, his mentoring style was reflected by those trainees, so that, for example, I use the same techniques and style as Earl in mentoring the people that came through my lab. In summary, Earl Davie was an amazing scientist and an amazing individual. His talents were recognized by the International Society of Thrombosis and Hemostasis when he was awarded the 1993 Distinguished Career Award. Earl really left a strong legacy for the science that he completed.